So good morning, sir, and all my classmates. I am Anandan Das, and I am going to discuss about endocrine regulation of molting in insects. So to start with the brief introduction, molting is a process in which animal sheds off or casts off its outer layer of either skin or any exoskeleton like hair, horns, or feathers, and replaces the same with a new one. So the process of molting can be seen in many animals, ranging from lower invertebrates to higher animals like mammals as well. So speaking specifically about molting in insects or in arthropods like crustaceans, we generally use the term called ecdysis. So ecdysis is nothing but the shedding of external skeleton or the exoskeleton of a arthropod for the purpose of growth, because as we know that exoskeleton is a hard and inflexible structure, so the growth of an insect gets restricted inside an exoskeleton. So for the purpose of its growth, it sheds off its exoskeleton at the end of its a developmental phase, and then it can regrow the exoskeleton again. So this complex process of ecdysis is mediated by a number of hormones which work collectively in a very organized fashion to serve their purpose and to drive home its purpose of ecdysis. So these are the hormones which are mainly responsible for the ecdysis process in insect. Starting with the eclosion hormone, there are ecdysis triggering hormone, pre-ecdysis triggering hormone, crustacean cardioactive peptide, corazonin, persicon, and the most importantly, the 20-hydroxyecdysone. So all the ecdysis and the hormonal control of ecdysis related experiments are generally conducted in a insect model called Menduca sexta, uh, uh, along with Menduca Drosophila and cockroach, there is periplanet, are also considered as experimental models. So here it is depicted a larval stage of Menduca where it is shown the different uh, regions of the body from which the hormones are being released. So the eclosion hormone and corazonin are being released from the neural cells present on the ventromedial region of the central nervous system of the insect. Then ETH and PETH are being released from the specialized endocrine cells called INCA cells. Then crustacean cardioactive peptide and barsicon are being released from a pair of neural cells called 27A and 704A cells present in the abdominal region of the central nervous system. And ultimately, the 20-hydroxyecdysone is being released by another pairs of endocrine cells called prothoracic gland. And these are the laser confocal micrographs of these glands and cells from which the hormones are being released. So all these hormones are actually peptide in nature except hydroxyecdysone, which is a steroid hormone. And the process of ecdysis is initiated by this hydroxyecdysone, which then stimulates the other concerned cells to secrete their respective hormones, so that the process of ecdysis, the various behavioral phases of ecdysis can be conducted. So all these hormones except ecdysis triggering hormone and hydroxyecdysone are being released by specific neural cells, but this ecdysis triggering hormone and hydroxyecdysone are being released by specialized endocrine cells, that is prothoracic gland and inca cells respectively. All these hormones have specific functions which will be discussed in some details in the subsequent slides. So the uh, the process of ecdysis is can be divided in broad heads in three different behavioral phases. These are pre-ecdysis behavioral phase, ecdysis behavioral phase, and post-ecdysis behavioral phase. So starting with the pre-ecdysis behavioral phase, so this is the phase where the outer skeleton or the cuticle of the insect loses its attachment with the muscle layer underneath the cell. There is by rhythmic compression and synchronous along with the abdomen and rhythmic retraction of the abdominal prolapse. This loosens its attachment to the external covering so that it can lose its outer cover. So this phase is directly regulated by the ecdysis triggering hormone and pre-ecdysis triggering hormone, but it is initiated by the 20-hydroxyecdysone, which initiates the Inca cells to release the ETH and PETH. It also further stimulates the corazonin, which in turn triggers ETH release from the Inca cell. So the decreasing level of hydroxyecdysone is actually responsible for initiating the behavioral phase. So this is a uh, complex circuitry of the ecdysis process, which is mediated by the hormones. So here there are two cases which can be considered. One case is in those insects which have a particular behavioral phase which is called ecdysis purge, and here in this sort of it, and the other behavioral phases of insect which do not have such thing called ecdysis purge. So ecdysis purge is nothing but a stimulus which helps this sort of insects to initiate the process of ecdysis. Although the process of ecdysis is mainly initiated by the declining level of ecdysone, but in some insects, ecdysis purge works as the limiting factor behind initiation process of ecdysis. So uh, sorry, what happens is when the ecdysone level gets decreased, it can directly initiate the process of ecdysis, and uh, it can initiate the ecdysis purge. So ecdysis purge is nothing but a stimulation in the insect in which the insect search for a specific substratum or a specific site to set it up before it can initiate the process of ecdysis. So some insects before initiating process of ecdysis search for a specific uh, place in which it can initiate the process. But in other sort of insects, the declining level of ecdysone is the only limiting factor behind initiation of ecdysis. So after ecdysis purge is being achieved, it stimulates the eclosion hormone neurons, which releases eclosion hormone and which further stimulates the Inca cell to release ETH. ETH in turn stimulates the eclosion hormone neurons as well. It works in a positive feedback manner, and then the Inca cells release ETH and PETH, which initiate the pre-ecdysis behavior, which can be further stimulated by the corazonin, which was earlier initiated by the declining level of ecdysone. So the only difference is that in this sort of behavior, the when the ecdysone level declines, the insect cannot help but to undergo ecdysis then and there only. But in this sort of behavior, the declining level of ecdysone initiates ecdysis purge, and then that ecdysis purge initiates the pre-ecdysis behavior. So now coming to the ecdysis phase, so this is the phase where the insect actually loses its skin and comes out of it completely. So this phase is actually controlled by directly controlled by CCAP. This is the crustacean cardioactive peptide, and as I've just mentioned that this crustacean cardioactive peptide is being released by 704A cells of the central nervous system, which is being stimulated by the eclosion hormone itself. So there is a concept called a ecdysis circuit delay, which helps in completion of the pre-ecdysis behavior before the initiation of ecdysis phase. So what happens is with with the synthesis of CCAP, there is another sort of compound called descending inhibitor, which is being synthesized by the similar sort of cells which synthesize the CCAP, and it is being stimulated by the EH itself. So what happens is the descending inhibitor in, inhibits the biological activity of CCAP for a while. So when the CCAP is being suppressed for the time being, the ecdysis behavior is being kept on hold. So while the ecdysis behavior is kept on hold, the remaining pre-ecdysis behavior can be completed in the meantime. So when that pre-ecdysis behavior is being completely done, or it is being completely um, uh, wrapped up, then the descending inhibitors loses its biological potency, and then CCAP can return back to its normal activity, and then it can start the process of ecdysis. So this is being demonstrated here in in a graph where we can see that the upper graph is for the descending inhibitors and the lower one is for the CCAP. Here we can see that both the graphs are are similar looking, that uh, both the descending inhibitors and the CCAP are being released in a simultaneous manner, but the difference is that the higher concentration of descending inhibitors have larger magnitude of inhibitory action over the uh, CCAP itself. It is there is a bigger dot, and by the course of time, when it its concentration or its potency gets lower and lower, the dots are getting smaller and smaller. That means the magnitude of its action over CCAP is getting reduced throughout the course of time. And when the descending inhibitors goes below its threshold level, that means it loses its activity, and then it has no then it can no longer uh, inhibit the CCAP. And but CCAP has still some of its concentration above the threshold level, and this remaining concentration of CCAP is the actual mediator of the ecdysis behavior, which can be seen in insects. So the difference is that the CCAP, when it is being released at the time when CCAP is released, but the action of CCAP is being elicited sometimes after, when the descending inhibitor has been completely removed from the circulatory system. So, so there it is that the ecdysis can initiate. So that is the process of the delay circuit which is being seen in ecdysis. So now coming to the final phase, which is the post-ecdysis behavior. It is the phase where the earlier processes are being completely done, and then the post-ecdysis behavior can initiate. The post-ecdysis behavior is nothing but the synthesis of the new exoskeleton, or the hardening of a new cuticle, and the formation of new wings, or the expansion of wings. So this phase is completely controlled by uh, enzyme called a hormone called bursicon. So bursicon is being released by the same set of cells which releases CCAP. So after, when the CCAP is being exhausted, then the bursicon activity can actually start, and then the post-ecdysis behavior can be initiated. So once in blood, bursicon not only activates tanning but also alters the physiology of the wing. So it also mediates the expansion of wings and the final few processes of ecdysis. So this is also mediated by cyclic AMP pathway and um, all these sort of things. So apart from the abdominal ganglion, that is the uh, it is the normal synthesis pathway for bursicon. Subesophageal portion of CNS also helps in release of bursicon. So to conclude, so here we have seen that a animal like um, small animal like insect also has such complex circuitry of hormonal regulation. So as already mentioned, that each phase of the insect hormonal control of ecdysis has specific set of enzymes, specific set of hormones which work in a collective fashion. So in pre-ecdysis phase, we have ETH and PETH as direct coordinators. In ecdysis phase we have a CCF is the direct coordinator, and at the post-ecdysis behavior we have Barsicon. But all these are collectively mediated by the 20-hydroxyecdysone, and apart from that, eclosion hormone and corazonin also has its effect. So the study of this sort of behavior in insect also helps us to get a bigger picture of the hormonal control that we see in the animal kingdom in general, and further research and emphasis will help us to know more about the subject. So these are some of the reference which I've used while making this presentation, and thank you very much.
Oh, you go to the table. Very nice presentation. You have one table, right?
Yes, sir.
What is corpora alata? Any idea?
Sorry, sir.
Corpora alata? You know that? No.
No, no, sir.
Okay, okay, it's fine. Nice presentation. Maybe you could not understand the theme behind the neuroendocrine system of insects.
Oh,
It's very complex, right?
yes, actually, it was a pretty new subject for us. We didn't study it actually earlier.
Yeah.
So,
I also find it difficult. This is very tough to me also, like you. So, uh, but you handled quite well. So,
thank you, sir.
and right now I am sending you one link. Hopefully, you have received all the link, right? So, are you using laptop?
Yes, sir.
Can you go to the link that I sent? Can you load the link if possible?
Yes, sir. Yes, sir. Please wait for a while. So, this is the link. Are you seeing it?
Could you scroll down?
Yes, sir.
Yeah, down, down, down. Go down. Yeah. Here crustacea comes. Class crustacea. Uh, it will take only 15 minutes of your time to read these notes. And after you read these notes, then we'll again consult in detail the neuroendocrine system of insect. But I'm including this uh, for your upcoming examination on 14th. Is this okay?
Okay, sir. Okay, sir.
In order to understand the basics of neuroendocrine system of insect, you need to at least read this 2-3 times, right?
Okay.
So today's presentation is over, and those who are from biochemistry don't leave, right? Be there. And next presentation, whose presentation? Raki,
Yes, sir.
uh, Binoy Sinha is there.
Yes, sir.
Binoy, could you give your next presentation?
Today,
No, tomorrow.
if possible, sir, Monday I can, sir.
Or you can do give on Monday. Then Raki tomorrow. Okay.
Okay, sir.
Raki will be okay tomorrow. Then neuroendocrine system Binoy Sinha on Monday.
Yes, sir.
And next will be mm, I'll let you know Ratnadeep maybe or Ramparanidas. On Monday, tomorrow Raki will give, then Binoy Sinha, then Ratnadeep or Ramparanidas. I'll let you know on Monday. Okay?